The structures of receptors are very diverse and include the following major categories, among others. Type 1. Ligand-gated ion channels. These receptors are typically the targets of fast neurotransmitters such as acetylcholine and GABA. Activation of these receptors results in changes in ion movement across a membrane. They have a heteromeric structure in that each subunit consists of the extracellular ligand binding domain and a transmembrane domain which includes 4 transmembrane alpha helices. The ligand binding cavities are located at the interface between the subunits. Type 2. G-protein coupled receptors. This is the largest family of receptors and includes the receptors for several hormones and slow transmitters, for example dopamine, metabotropic glutamate. They are composed of 7 transmembrane alpha helices. The loops connecting the alpha helices form extracellular and intracellular domains. The binding site for larger peptide ligands is usually located in the extracellular domain, whereas the binding site for smaller non-peptide ligands is often located between the 7 alpha helices and 1 extracellular loop. The aforementioned receptors are coupled to different intracellular effector systems via G-proteins. G-proteins are heterotrimers made up of 3 subunits, alpha, beta, and gamma. In the inactive state, the 3 subunits associate together and the alpha subunit binds GDP. G-protein activation causes a conformational change, which leads to the exchange of GDP for GTP. GTP binding to the alpha subunit causes dissociation of the beta and gamma subunits. Furthermore, the 3 subunits, alpha, beta, and gamma have additional 4 main classes based on their primary sequence. These include Gs, D, GQ and G12. Type 3. Kinase-linked and related receptors. They are composed of an extracellular domain containing the ligand binding site and an intracellular domain, often with enzymatic function, linked by a single transmembrane alpha helix. The insulin receptor is an example. Type 4. Nuclear receptors. While they are called nuclear receptors, they are actually located in the cytoplasm and migrate to the nucleus after binding with their ligands. They are composed of a C-terminal ligand binding region, a core DNA binding domain and an N-terminal domain that contains the AF1 region. The core region has 2 zinc fingers that are responsible for recognizing the DNA sequences specific to this receptor. The N-terminus interacts with other cellular transcription factors in a ligand-independent manner, and, depending on these interactions, it can modify the binding/activity of the receptor. Steroid and thyroid hormone receptors are examples of such receptors. Membrane receptors may be isolated from cell membranes by complex extraction procedures using solvents, detergents, and/or affinity purification. The structures and actions of receptors may be studied by using biophysical methods such as X-ray crystallography, NMR, circular dichroism, and dual polarization interferometry. Computer simulations of the dynamic behavior of receptors have been used to gain understanding of their mechanisms of action.